Yep. Okay, we'll see if that works out.
I'll definitely
Yeah,
put that proposition to them.
right. Wonderful.
Okay, so now it's 10 to, so let's begin. Please go ahead, Sandy.
Okay, well, thanks, Philip, for organising this meeting. I'm really sorry that I missed essentially all of it, and I'm just sort of jumping in for the talk, but I will try and stick around for the discussion. But today I'm going to talk to you a little bit about how we can use sort of mechanistic models versus machine learning sort of models in really trying to make decisions about when cancer therapy should be given. And for those of you that are sci-fi fans, you might recognize this little graphic here from 2001: A Space Odyssey, which is a classic movie where man and machine end up battling one another. And that's HAL on the right-hand side. If you've not seen that movie, you should definitely go see it. So a little bit of context. Philip said, if you sort of joined a few minutes ago, that I run the Integrated Mathematical Oncology Department, and it's a very kind of rich and diverse group of faculty and postdocs and staff. And the goal, really our main focus, is to better understand, predict, and treat cancer. And the way we do that is really through the integration of experimental approaches, clinical application, all sort of tied together with mathematical modeling. And at Moffitt, we've been very fortunate to have quite a key player in the experimental world, someone who's also an expert on imaging, Bob Gillies, and many of our projects integrate people from his lab who are sort of testing out and sort of implementing a lot of the experimental approaches, but it's not exclusively him. And then Bob Gattenby, who's actually a radiologist in training, and he's one that's been in some sense the clinical translator that helps push some of our theoretical ideas into the clinic. So within my department, we have something like 10 clinical trials that are being driven by mathematical models to make treatment decisions. And some of those trials are sort of focused on what we call adaptive therapy, but many, another, at least 50% of them aren't. So I thought it would be worth briefly giving you a little bit of context about my view on cancer. And I want to emphasize this definitely isn't the standard view from a cancer biologist's point, but I think it'll probably jive with most of the modelers here. So cancer is an emergent phenomena. So you've probably heard that cancer is a disease of mutations, but mutations are necessary but not sufficient actually to drive cancer. And a great example of that is most of you probably have moles at some place in your body, and moles are actually, they contain all the mutations that you would need to have cancer, but they don't turn into cancer. Right, and so it's not just enough to have the mutations. Now, of course, some moles do turn into cancer, but not all of them do. And so one of the reasons for that is that we think that the kind of niche and the kind of homeostatic regulation of the tissue of the skin, of the architecture of the skin, really regulates the form and function of those cells, even if they're rogue. Because they're not all rogue, many of them are still behaving normally. And so, our systems that make up our organs and so on really have a lot of built-in redundancy and robustness to perturbations. So, huge wounding events or damage that can happen, and then your system repairs itself, and it does so in quite an efficient way. It might not be the exact same as it was, but what emerges is still a functioning system that is quite robust. One of the issues with that kind of emergent aspect of cancer is how do you actually represent that experimentally? So how do we do experiments that really capture the robustness and redundancy of normal tissues? And a lot of experimentalists resort to animal models as a way to sort of try and do that. But of course, when you're putting a human cell in an animal, you're merging two very distinct systems, but it's at least capturing some of that niche, but it's not quite the same thing. But still, that's a step in the right direction. So really, over the decades of cancer research, I think what's come clear is that we really understand many of the key processes that drive cancer initiation and progression, and also that drive cancer responses to treatment. But many of those processes are understood in isolation. Like there's a tendency to focus on little components of that pathway, of that system, and understand it in detail. But of course, as modelers, we think systemically and we think about the pieces of the puzzle together. And so how do we link the individual processes to drive the outcome, the emergent outcome? And that's where models really are kind of, excellent tool for this. And again, coming back to homeostasis is hard to capture. And, even modelers have a tendency to sort of forget that we need to have a system that should act like it doesn't have cancer when it doesn't, and then only behave like it's, cancer is there when you add cancer. And so that one of the emergent aspects is, of course, the dynamics of the process of cancer growing, of cancer invading, and of cancer responding to treatment. And that dynamic aspect is one of the potential ways that we can connect models with data, especially sparse data, when you've got limited access to, say, a patient only through imaging or through serum markers. So one of the problems that we've been working on for a number of years now is really trying to think of strategies to treat patients who have metastatic cancer. And this is a scan, it's a whole body scan of a patient with metastatic melanoma, which is a skin cancer. And the brain and the bladder there, which are bright red, are okay, they should be red, but the rest of the torso below the lungs and the abdomen, all of that red stuff there shouldn't be there. And that's all red because effectively the cancer is incredibly glycolytic. It's using a lot of glucose and it really lights up on the scan. And if you saw the patient's skin, they would have huge black sort of lesions all over their body. It looks horrible. And this doesn't look much better. But what's been kind of amazing is in the last, really it's only been in the last decade, it's not even been a decade, new drugs have been developed that specifically target the mutational aspects of this cancer. And so this patient had a BRAF mutant melanoma. It's a specific mutation that occurs in those cancers. And so there's been a drug that would go and target and inhibit cancer cells from dividing that have that mutation. And by giving the patient that drug, a mere 2 months later, it's literally less than 8 weeks, the patient looks essentially cured. Like if you saw the surface of their skin, their body completely looked normal, they look healthier. But as these scans reveal, there's still some small kind of metabolically active lesions there that are showing you that not all the cancer has been eradicated. So why is that? Right? Why is it not eradicating everything? And the reason is that not all of the tumor cells are the same. Not all of them have that mutation, or if they have that mutation, they've got another mutation that lets them escape the potential impact of that drug. And so unfortunately for this patient, within 6 months of receiving that great miraculous response, they then go on to have progression, and now they're resistant to that drug. And essentially they fail that therapy, and that really means that they're on a sort of trajectory to really die. There's not much we can do. There's still multiple sort of treatments we can try, but ultimately it's a losing battle. So this is called a targeted therapy, and they fail because of drug resistance. So is there something different that we can do? So the reason why targeted therapies fail is because, of course, the resistant population is more than likely already present, but it's also because we give it the maximum tolerated dose. And so if we just look at the little cartoon at the top there, if we imagine the tumor consists of many different cellular populations who have different mutations. And of course, in that image that I showed you, there was multiple small tumors throughout the body. If I then color the cells according to how sensitive or resistant they are, and I give maximum tolerated dose, then what happens is I end up with eradicating all of the cells that are sensitive to the disease and leaving behind only those that are resistant. And that's the pink cells in this cartoon. If I continue to hold on the drug now, I'm wasting my time with that drug because there's nothing there else to kill. There's nothing else going to be responsive. And so a number of years ago, Bob Gattenby came up with an idea really that was kind of driven by pest management for crops. And that is, in crops, they don't spray the entire field with pesticide because very rapidly they get resistance. What they do is only spray, like say, 3/4 of the field, and they leave a quarter unexposed. And that quarter unexposed, you're essentially giving up to the pests. But what happens is that the rest of the crop you're killing those pests, and then next season, that quarter that survived repopulates the rest of the field, and you can then redo this process again and again. And so in the lower picture, what you see is same sort of tumor, but I'm now going to give what we call the minimum effective dose. So I kill it a bit, but not completely. And I leave some of those sensitive cells behind. And now I stop treatment altogether. And when I stop treatment, the tumor grows back. And what's important here is that not all of the cells are equally fit. Some of them grow back quicker than the others. And because we think there's a high probability that resistance isn't free, resistance has a cost, that means when you stop treatment, resistant cells grow slower than their sensitive counterparts. And so now what I can do is repeat this process below many times and potentially control cancer for longer. So it's a strategy of control rather than eradication. But of course, it relies on this assumption that resistance may have a cost. And so here's some experimental data and some standard sort of cell lines. So MCF7 is a breast cancer cell line, and doxorubicin is a drug that you typically treat breast cancer with. And so in red on the left there is the MCF7 doxorubicin resistant cell line, and in green is its sensitive counterpart. And if you co-culture them together in sort of, in this case, it's actually low glucose conditions, what happens is very quickly the sensitive cells outcompete the resistant ones. On the right-hand side, there's now, I'm comparing metastatic melanoma cell lines. And if we look at the WM164 and its resistant counterpart, and this is resistant to that BRAF inhibitor, you see that the black sensitive population grows quicker than its resistant counterpart. However, if we compare this 1205LU population, you can see that the sensitive and resistant are equally as fit. So it's possible that some cancers will pay a cost and some won't. Another thing that sort of emerged from this is, of course, is resistance pre-existing, or is it something that emerges through mutation, or is it emerging through some other mechanism like a switch to a different pathway or epigenetic? And is it actually a plastic behavior, meaning can I become resistant to a treatment and then can I be resensitized to that treatment? And this experiment here, this data I'm showing you, if you just focus on the left-hand side, this shows a process of a cancer cell line becoming resistant and then returning to sensitivity. So the black line there is a dose response curve to this BRAF inhibitor. And you can see that you're getting this kind of log kill and then it sort of tails off and in black. And the resistant counterpart to that is the dark blue line, right? So these are run with many replicants. Now what we do is we take the drug off the cell line for a week, and then we test its sensitivity then for 2 weeks, then for 3 weeks, and so on. What you can see is by about week 5 or 6, you've got, you're not really moving any further. You're not coming close to being fully sensitive again to that original population. So it implies there's definitely some return to sensitivity over time, but it doesn't go all the way. If you compare that with the 1205LU cell line, what you can see is that when I go from being black sensitive to the blue resistant, after that I never actually fully return to being sensitive again, right? So essentially there's no shift It's almost like resistance is actually a permanent behavior. It's not plastic in that cell line. And it's interesting that that cell line also didn't appear to pay a cost for being resistant. Another thing that we think could be important is turnover. And I'm sort of going through this at a fairly quick step because I want to get to the focus, but I think it's useful to give you this background. But this shows a cellular automaton model. It's the same initial conditions in each situation. And what I'm going to show you on the left-hand side, green cells are sensitive, red cells are resistant. I'm going to give just drug continuously, and the little white square shows you when the drug is on. In the middle, I'm going to do this adaptive therapy that I was telling you about, which is I'm going to watch the tumor shrink to 50% of its size, and then I'm going to stop the treatment. And then when the tumor goes back to its original size, I'll start the treatment again. And on the far right-hand side, what I'm going to do is essentially have random death in addition to that. So turnover, just random kill of all cells at a certain rate. So when I do that, what you can see is on the left-hand side, very quickly, you select for resistance. In the middle, we had a little bit of an oscillation, and then very quickly, it kind of heads to resistance. And if you look below, it shows the kind of curves, the dynamics of that. And on the right-hand side, what you can see is we get control for a bit longer before you fail. And so that shows you that turnover can, in fact, have a big impact on, in this case, how quickly you get to this sort of original level, actually 20% above that level, which is a kind of typical resist failure. So I'll talk now specifically about some animal work before we move on to talking about a different type of cancer. And this animal work is in metastatic melanoma. And that's that WM164 cell line I've been telling you about. And what you can see in all these little graphs here is essentially tumor cell line that was engrafted into a mouse and then tracked over time. So the far left-hand side shows the doubling, the growth of that for multiple mice. So each is the same experiment, but you're just doing it with different mice. And if you look at no treatment versus continuous, what you see is no treatment, they all grow, but they grow at different rates. Under continuous treatment, what you can see is almost by around day 10, you see some sort of suppression, but almost all of them fail the treatment and become resistant, right? Those cell lines were sensitive at the beginning of the treatment. And then we do some intermittent dosing where we say 2 days on treatment, 6 days off, and so on. And what's kind of interesting here to me is that when you do the treatment on and off, that kind of diversity of response that you saw for no treatment and continuous treatment actually narrows, somehow sort of unifies the dynamics of the population. And so that data then was used to drive a mathematical model. And this mathematical model, based on the understanding that we had that the WM164 cell line was essentially a plastic cell line, it was one that when treatment's off would return to sensitivity, we came up with a very simple coupled model of differential equations that take a sensitive population and under treatment make it resistant, and out of treatment make it return to sensitivity. And so this sort of, essentially switching model, this very simple switching model is what we came up with. So taking that model now and fitting it to that data that I just showed you, you can actually get reasonably good fits to all of those mice. And so when we're doing this sort of fit, we take the entire collection of mice and just try and fit them all simultaneously. And so, if you look closely, you'll find some areas where it doesn't fit so well, but the bulk of it does a pretty good job. So with that calibrated model, we then took a new set of mice and we used that model to make treatment decisions. And so what we did was, and in each line here is a mouse that's now been treated by the mathematical model. So we gave each mouse 2 weeks of treatment up front, and then we used the model based on the dynamics of that individual mouse's response to treatment, whether to decide after we stop the treatment, we say, when should the treatment come back on? And what you can see is that the black line after that first red varies for some of the mice. So you can see that, for example, mouse 15 and 22 don't start again till 2 weeks later. Mouse 8 only starts again briefly and then stops again. And then there's a collection down the bottom that all seem to sort of come back on 2 weeks later. What's important, though, is that the actual dynamics of these mice, even if the timing of their treatment might look similar, is the volume change in their tumor is quite different. That's what the V is. And you can see that, you know, some mice even have a volume 8-fold change. This one's almost 8. Some of them barely change at all. And so essentially, this approach that we then came up with, we can compare with continuous versus intermittent, fixed intermittent schedule versus an intermittent schedule that the model said would be best, and that was the 8 days on, 4 days off. And what you can see is that the adaptive approach statistically is better. But what's clear there is we're not curing these mice, right? We're just controlling it for a bit longer. And that is really kind of, in some sense, the strategy here for using this approach. Okay, so let me now talk about where we're going to bring the kind of man versus machine, or mechanistic modeling versus machine learning, and that's in ovarian cancer. So ovarian cancer is one of the major cancers for women. Most of those cancers occur in the area that's called the omentum, and that sort of shows you a little bit of the biology there. And they can be fairly small, but when they metastasize, they kind of metastasize locally all throughout the omentum. And so PARP inhibitor treatment is really kind of one of the major standards of treatment now for women in the US. And this graph on the right, which I can't actually see, I need to move that, shows you essentially what happens when we give that treatment. Initially, you get some response, then a kind of almost like, you know, control period, and then progression, right? So it takes some time, but effectively you get resistance. So could we do something better than just maximum tolerated dose? And so what we wanted to do was build a mechanistic model to think about that. But at the time, I also had a summer student who was very interested in machine learning. And we got talking about, well, perhaps we could also build a machine learning model, because we had this data that I'm going to show you that could help train it. And so one of the things that we then debated about were the pros and cons of the different approaches. And I thought, this has probably already been discussed, but and I'm definitely not an expert in this, but I'll share what we kind of concluded. So, you know, I think machine learning is potentially useful for uncovering patterns that are often unseen by human observers, or like you might miss based on that subtlety, but if there's enough data, you'll see the patterns. They're also particularly good at dealing with noise. And so one of the things that you saw in that animal data set is that there's quite a bit of variability, right? Now, how much of that's actually based on the injection technique and the mouse itself is unclear. But you also get very noisy data, stochastic aspects of it. They're often difficult to interpret biologically because they tend to be black boxes where you can't see why it does what it does. And often small changes to the input can make them break, right? So they can struggle with robustness. And of course, they require large data. On the flip, mechanistic models explicitly incorporate known biology. They can build testable hypotheses without large data sets, and they're particularly good at capturing temporal dynamics. However, the kind of flip of what that constraints are that you've kind of dictated what biology you want to put in there, and you may be too focused on the kind of single phenomena, right? You've already almost built in what you think is the important aspect. And these sorts of models can sometimes struggle with noisy and diverse data. And of course, you're going to miss those patterns that might be present that would be subtly seen by machine learning. So this is some of the data that we used. So what you can see here in these kind of almost tiny little signatures are the results of many three-dimensional spheroid sort of organoid experiments where what we're doing is we're taking varying cancer cell lines and growing them in 3D culture and then treating them or not treating them, which is control, no treatment, giving them maximum tolerated dose, and then giving them a combination of different treatments. And here there's the kind of blue and the red are actually two different cell lines. But what you can see, and you're not meant to be able to read this very clearly, but it was just to show you there's quite a lot of data here. And there's like every sort of little snapshot in time is an image. And so we have like 60,000 images, 192 spheroids, and they're all growing under different treatments, right? And so there's 3 different adaptive therapy approaches there. It doesn't matter what they are. The point is there's a huge data set. And the data is essentially looks like in reality what you're seeing on the left-hand side, which is this kind of imaging of a blob that's changing as it's treated, right? And so we've got to turn that into some sort of usable data. Now, I know that there's many applications to machine learning which will take images and sort of pull out features, or even will kind of find patterns in images without any kind of prior knowledge. But here we were interested in actually trying to get some sort of size metric out of these images. So some very simple image analysis here was used to essentially extract area, perimeter, radius, X variance, and Y variance from these data. And what you can see there is that, you know, it's a bit crude. It's not a great way of doing this, so it could be done better, but we do this consistently across all of the data. So you're going to get some consistency, at least in that. So given that data, then what we have, or what emerges from that, is a set of what we can call control state variables. So that's time since, you know, essentially the cancer it was initiated, time since the drug dose, whether the drug was given or not, and what the actual dose of the drug is. And so you can see on the right hand side, for example, in control, there's 0. This is showing you the drug dose, 0 drug, MTD, it was 10, and so on. So we take that and now we're going to implement it. And here it's a feed forward network, deep neural network. And that architecture was chosen because, well, the students seemed to think that was the best way to sort of tackle this. Of course, there's potentially other ways to do that. But one of the things that's kind of important here is that we're then, you know, predicting with this network the area, perimeter, radius, X variance, Y variance, and we're training it with all of that data that I just showed you. And so once you have such a trained network, what we're then going to do is take the output of that network and feed it back into itself. And that will allow us then to potentially predict the next, you know, treatment outcome. And we can do this. And so basically you can essentially do, you know, multiple time steps that you're going to feed in to this model and then make it predict the next outcome. So for example, is it more predictive? Is it better for this network to have as an input just the immediate previous step in its current state, or say the last 3 steps in its current state, right? And so what I'm showing you here is how predictive this is, and this is a kind of complicated figure, but if you just look on the left-hand side for a second, each little box there is one run of the experiment. So it's one spheroid that we're watching. So the blue here is, I think the blue here is the real data, and the red is the machine learned data. And what we're doing on the left is we're sort of saying, here's your input, tell me the next day. So each little point is then driving its immediate next point prediction. And it's really quite good at predicting the immediate next step, right? But that's kind of probably not too surprising because we trained it on that data. And versus on the right-hand side, what we're doing is we're saying, we'll give you, in this case, 3 previous time steps, now predict the entire time going forward, predict the entire progression. And it doesn't do as well, and I'm showing you there the blue sort of predicted, not red predicted, sorry, and then a variance around that, just to give you a feel for what it is. And you can see that it, you know, it is doing something, but it's definitely missing the mark in some cases of the dynamics in between, but it kind of gets the start and the end pretty well. So it doesn't do so well at predicting too far into the future. So with that trained network, we then looked at the policy that came out of it and just tried to understand what it was telling us, right? You know, why is it doing what it's doing? And what we realized, because we could, of course, systematically have different inputs and see what it predicts to do, is that it's found that large tumors need a vacation. They need a treatment holiday between pretty high doses, whereas smaller tumors need more maximum tolerated dose treatments. And that's, I should have said that one of the things that we did with this trained network was we then did a new set of experiments where we used this network to make treatment predictions. How am I doing for time, Philip? I can't hear you, but
roughly 10 minutes.
Okay, good. So what you can see there is, and if you look at in the small tumor one, for example, time here is, you know, given the first treatment, the next treatment what will that be, and so on. And so what this for the smaller tumor is saying is that give the maximum dose for the first treatment, essentially give the maximum dose for the second treatment, and give a slightly lower dose for the third, and so on. Whereas if you contrast that with a large tumor, you see give a kind of maximum dose for the first treatment, give 2 holiday doses, then give a kind of mixed dose for the next treatment. And so that's what this machine learning model pulled out from all of that data. And so that was, oh yeah, and there's also, we can sort of look at essentially how does this maximum sort of tolerated dose behavior drive it's essentially back propagating through the network. We can talk about that later. But essentially, moving on to the mechanistic modeling now, I showed you one specific model, which was a kind of switching model. What we wanted to do here was really come up with something that was a bit more robust than just a single model to try and make treatment decisions, because we're not sure we actually have the right model. And so we came up with this model ensemble framework. And essentially, this shows the kind of pipeline that I'm going to apply, which is, of course, way to extract that data and process it. And I showed you a kind of little bit already how that was done. We fit then our models. We then optimize the treatment. So we make predictions forward in time, and then we make a decision based on the kind of ensemble, like how the group decides to treat, right? And the idea is that maybe we can sort of deal with the diversity or with the poorness of fit that might occur between, or the disagreement between the models. So we chose 6 different models. There's 2 kind of exponential models, 2 logistic, and 2 sort of surface growth models. And if you know your growth models, you'll recognize many of these players. And this is something that Philip is all too familiar with. He wrote this beautiful model paper, which is something that we talk about a lot. But anyway, with these different models, you're making different assumptions in some sense about the biology. So in a way, we're trying to, let's say, compensate for the built-in assumptions by giving a diversity of approaches. But they're still all assuming a well-mixed system, and they're still all assuming that there's sensitive and resistant populations, but some of them actually have switching in them, okay? And of course, there's different degrees and numbers of parameters. So we took that model, and so this kind of just shows a little bit of the pipeline. I kind of showed you how we explore, because one of the reasons I think it's important to talk about this is that we don't just get gifted the data to make a decision. So on the day that you're trying to make a treatment decision, you've got to extract the images, transfer those files, segment them, and first of all, you want to double check the segmentation. That gives you then these kind of growth functions, which you can then try and fit your models to. And of course, there's many ways to fit, and you're trying to do that in a robust way. So you're going to get some bits that are bad and some bits that are good. And so we chose a sort of 80-20 rule for that. And now we take the fitted models and we predict them forward in time, and we simulate many sort of, let's say candidate runs for each model. And then we take from that essentially a kind of treatment versus no treatment option. And we say, well, what's the decision? Do 4 of the 6 models say treat and 2 of them say vacation? And then depending on who has the upper hand there, we treat or we don't treat. And this just shows you kind of weaken the life of, and this was Maxi Strobel, who was the student that was doing this. This is what he had to do. And so the processing of the data is the most consuming aspect here. So even though we get the data on Thursday, we don't actually get the data, the process data until the Sunday. And then you're sort of applying that decision on the Monday. So it's almost like 4 days after they've already changed in the process. So there's a bit of a delay. I don't know what's going on there. Anyway, there was something there. So basically this bottleneck was a bit of an issue. But of course, for the machine learning model, as soon as we fed it the input, it instantly gave us a response on what it should do, right? So it was really efficient, it was fast. Unlike the mathematical models where we did this whole sort of, dancing with fits and so on. So what does it look like? So here's a specific example. And so what you can see here is the data that's been pulled out of these this analysis, so the crosses. And then the color is when the treatment's on, purple and white when the treatment's off. And so this was showing you just little snapshots of what those images look like. And so we're at, say, 0.4, we've now got to make a decision. And so the current point there was already on treatment. And now when we kind of do our ensemble of models, we actually got all 6 models to agree that treatment should remain on. So in this case, it was actually quite a consensus, everybody agreed. So I know you're desperate to know who won. Was it the machine learning model, or was it the mechanistic ensemble? But let me show you how the machine learning model did in comparison to the other treatments that were in there. So control is no treatment. So one would hope it would do better than no treatment, right? And so if you look at the upper panel there, blue is our neural network model and red is no treatment. And what you can see is, yes, blue certainly does better. It's a bit noisy. There's many replicates in here. If we compare it with maximum tolerated dose, blue still appears to do better, but one would argue that this is not statistically significant. And if we then use it, and this is one of our adaptive therapies, I still think the machine learning model does better. Now, this is what you've all been waiting for. Here is the mechanistic framework in red and the machine learning model in blue. And yes, the machine learning model did better. So on top of all of that, the fact that even though we had this sort of redundancy and so on built into these mechanistic models, it's sort of impressive that the machine learning model still managed to find a kind of policy that pushed forward and did better. And I think one of the reasons for this personally is that these are noisy data. The spheroids are very, very stochastic. And you can see that the error bars are huge. And in particular, when you compare the ODE error bars to the machine learning model, it's almost like the machine learning policy drove less variance in the tumors for whatever reason. Okay, so let me conclude. One of the things that we talked about was adaptive therapy. And really what the heart of that is like saying that when you treat a patient, don't ignore their response to that treatment. Change your treatment in response to what their tumor is doing. So really you're saying, let's see how to control their disease based on how they're responding. Don't wait until they become resistant to the treatment. Now, of course, this generally is only really applying to metastatic disease that we're talking about, because we're actually pretty darn good at treating primary disease with maximum tolerated dose strategies, right? So this is really more about end of the line sort of treatments. And I thought, given the focus of this workshop, I'd reflect a little bit on how could mechanistic and machine models, integrate or relate to one another. And you're probably going to have that to take the discussion, but for my tuppence, I think there's a huge potential here. I think what's clear is mechanistic models seem to propagate time well, and I think there probably is classes of machine learning model that do that well too. We found that when we went back to really 3 days prior to feed anything more than 3 days into that network, we gained nothing. It didn't give us any more better fit, right? But of course, as far as the machine learning model is concerned, time is just other data. It's not time in the way that we consider it in the mechanistic model, right? I think that the noise that we saw, the real variance that we saw in those spheroids, the machine learning model seemed to handle that much better than the mechanistic model that was looking for the best fit, right? So, I think there's some options there, which is, could we integrate the two to make a simple prediction? Could we just add the machine learning model to the ensemble and then it becomes another player? Or could it be that we actually learn from that machine learning model? So one of the things that we learned is that it was treating large tumors and small tumors very differently. So does that mean that we actually need to build models that treat large tumors in a different way? Is there some sort of size dependence or density dependence that we're missing in those models? And that needs to be integrated, because the machine learning approach seemed to pull that out. Another is that could the kind of temporal dynamics from mechanistic models be integrated into the machine learning model itself, so that it's actually, you've got a sort of piece that's coming from the data and a piece that's coming from the mechanistic models that then are integrated in the machine learning algorithm itself. And so I'm not sure how we do that, but one could imagine that it could help sort of refine or retrain the network as time went on. So anyway, that's just a few thoughts on this. But I'd like to really specifically shout out those that did all the work. So the kind of early melanoma work that I discussed, all the modeling was done by Eunjun Kim. The kind of experimental side was done by Ina Smalley and Kieran Smalley at Moffitt. The machine learning stuff was all done by Matt Wicker, who's a PhD student at Oxford and has now spends most of his time developing self-driving car software stuff. And Maxi Strobel did the kind of model ensemble and has been doing a lot of the adaptive therapy work. And he's been co-supervised by Philip Maney at Oxford. And then Mehdi Noumanji, who's in Bob Gillies' lab, did all the kind of ovarian spheroid work. So thanks everyone. Hopefully it was relevant and not too biological for you all.
Thanks a lot, Sandy, for a nice talk. Do we have any questions from the audience? Well, I have one question. So the framework that was built around the mechanistic models was quite complicated, as far as I understood. So there are probably a lot of parameters in there that could somehow change the result. And would it be possible to do this, I don't know, to build that framework maybe in a more machine learning fashion? Or, I mean, because you have all these sort of hyperparameters that you maybe want to adjust.
Exactly.
Yeah.
So I think that the way that we treated that, let's say we had this sort of 6 individual fits, right, for each model. How does then one make a decision with that? We took a very naive way to do that, which was basically, binary decisions for each model, and then the one with the most decisions wins. Could we have weighted it? Could we have thought differently about the importance of each individual model and its fit, and then weighted its decision differently.
Yeah.
Right, so that would be some, but that's adding more complexity. I think that the framework itself isn't that sophisticated. I think having 6 models is quite complicated for sure, and they all have individual parameters, and some of them have way more parameters than others. But part of the motivation for doing that is that when we used a single model, because we had some, obviously we played with this beforehand, it wasn't as good. It just didn't do as good a job because, of course, it was fallen over because of the stochasticity variance that we're seeing in the data. So if it had been a much cleaner data set, a single model would have worked fine. And I think this does talk to your point, Philip, about could we then use machine learning in a way to help choose which of those models should be the decider, or how weight those models in some sense to make a decision.
Yeah. So yeah, and one comment then also in my talk, I mentioned a recent paper by Ruth Baker and Matt Simpson where they use a neural network to encode a differential equation.
Yeah.
So maybe that would be an interesting approach to use as well, or maybe it would have to maybe have some sort of stochastic element as well. But I mean, yeah, I think that would be an interesting application in this case.
Yeah, sort of back calculate the model that you're going to use to then fit the data to.
Exactly. Yeah. Somehow, something like that. Okay. So I think Torbjörn had a question.
Right. Thank you very much, Sandy, for this. So I have a question at the end there. When you announced the winner, the measure you had was how well it made the predictions, right?
I know it wasn't. The measure was how well it controlled the tumor.
Yeah. Okay. Yeah.
On the ensemble, okay. You mean?
Yeah, to make it as small as possible in that model. Right. Yeah. Okay. But okay. That was the only question I had. Okay, but I thought it was more of an error thing. So we need also to compare this with reality in some sense as well, at some point.
Yeah. So the the winner in the ensemble made the decision, right, for the mechanistic models, but the winner of the different approaches was who kept the tumor smallest for the across all the replicates.
Right.
At the end of the day, and so there was 6 different treatment strategies plus mechanistic modeling plus the machine learning, right, and some of those other strategies did okay too, but comparing mechanistic versus machine learning for this data set, and then for those experiments that those models drove afterwards, mechanistic modeling didn't do as well as machine learning.
So I guess in order to make these kind of controlled in an optimal way, you need to have really good predictions as a first step, right? To be able to choose the optimal way. So in that sense, maybe the machine learning algorithm also made better predictions to be able to combine.
Yeah, so I think that's one of the points I was showing you. I don't know if I think it's just. Oh yeah, that was, I forgot to show you that. That was my actually the future's really man and machine.
Yeah, exactly. Yeah, maybe that's a good point too.
Yes, but I was going to show you just this part here, because that's exactly where I think machine learning models do seem to be incredibly good at predicting the immediate next point, right? And if you think about it, that's essentially what we're asking of these models was, given the current state of the system and say the 3 previous states, what's the next one and what should I do, right? The immediate next one. It didn't do so well at predicting the end, right? But the end, so that's like the immediate short-term prediction on the left and the kind of long-term prediction on the right. But you're not asking it to make a long-term prediction when you're making a treatment decision. It's immediate next step.
Yeah.
So I think the machine learning model was really good at that, right?
Good. Thanks.
Good. Do we have any other questions?
Yeah, if I may.
Yep.
Hi, my name is Fred Vermolen. I'm from the Hasselt University in Belgium. So thank you very much for your talk, Alexander. It was a very nice talk and I enjoyed it very much. It was very interesting. And so the only question I have is that, so you have been dealing with these mechanistic models. And so these models are based on ordinary differential equations. Do you have any expertise or any comparison with mechanistic models that are based on partial differential equations? Because this type of models could also involve spatial aspects of the tumor. Yeah, and this could also be an additional, could add an additional value to the approximation of how the tumor is developing over time.
Yeah, that's actually an excellent point. And we do have models that look at spatial aspects, both in terms of partial differential equations, but also in terms of sort of agent-based approaches. But your point's well taken in this specific result, because I think that the machine learning model did well because it could differentiate small tumors with large variants versus large tumors with small variants, right? So that's a spatial aspect. And the kind of ODE model would just treat this as different numbers of cells, right? And so if I had space in there, I could potentially capture the sort of spatial spread difference. So in terms of small tumors, they could potentially contain large numbers of cells, even though they're small, right? Versus them being much more spread out in the large tumor. So I think that the spatial architecture could in fact be quite important. And I only showed you a couple of images. And one of the things that happens as you treat over time is you get fragmentation of the tumor masses, right? So if you're treating it as one big lump, as opposed to 6 smaller lumps, then you can see potentially you're going to get different dynamics, right? So I think that potentially could have done a better job. But, I think this comes back to also Philip's point, which is this framework is already quite complicated, and adding PDEs is going to add a huge other variable, which is space. In this case, I presume that's the partial part you were thinking about, the space. And, that's another complicated thing to try and fit here, right? So, but however, one could fit it in a kind of, let's say, crude way, like to do with the spread, whether it was, more focused or if it was more spread. And we actually thought about, in some sense, not building a PDE, but building a compartmentalized ODE where we had 2 different populations that were kind of inner versus outer, right? Edge versus core, to try and capture this phenomenon. So it's almost like pseudo space. So.
Okay, good. I think I'll hand over to Torbjörn, who will lead the discussion now.
Thank you. So thank you everyone for this great workshop. We have still some minutes here, 17 minutes for a discussion together. I thought I was going to set the table a bit by just recalling what we were discussing. Can you see that presentation now? Starting with Bernd's questions. I think there were like 3 different questions. It's always hard to have the person who said it in the audience and then try to reformulate it. But I think the underlying message was that is mathematical modeling obsolete now, or could it still be useful, or do we face the combo as the best way to move forward? I just want to add something from my previous thing, that to start with maybe first question here, that I was in Stanford and saw a talk by Andrew Ng, and he talked about the 2 greatest challenges in AI, and that was a lack of data and the need of better architectures. And the 1 year after that, I saw the first front page here at New York Times, and they pointed out the sweatshops. We've been talking about sweat in this seminar, with another sweatshop here, when a lot of underpaid workers were annotating images of colon cancer, that they thought that was a really good, vacation from the usual beheading or sex things that they have to do. So the cost of creating new annotated data is increasing, and the low-hanging fruits are already taken. And this is a sketch that Andrew made about we need better architectures. And to me, it sounded like we need to go back and making models to help us make better machine learning. And that is what Philip brought in light here, that Ruth Baker's last paper, or the latest paper, Ruth Baker here, about using machine learning in combination with PDEs to create better PD model. And that was the toolbox also of Robert when he talked about this combination as well, pointing out, for example, E.A. Feynman, and later we heard about the Quantic project, I think, but from Sebastian. And the toolbox was also basically saved here in High-Tech's talk. And he talked about Rudin, which famous mathematician name. I don't know if they're related, but she presented this kind of diagram of effectiveness of explanation in one end and prediction on the other axis. And that reminds us of another table or another picture that Philip and I had in a little book, but we had the predicting power on the other axis instead. And so, and the statement was that maybe there is like a negative correlation, but we talked more about the dynamics, that models are evolving and getting more and more like black boxes if you add more and more details. Anyway, so complexity versus simplicity. Who is the winner? And that was a question that was also addressed by Ayla, having different ABC models for fitting the sweat gland predictions here. And we have other models here that talking about mechanistic model versus machine learning, and some really nice one-liners about when machine learning is not making the really promising statement that they have been in this maybe a bit overheated situation. And also and a recipe, we got that recipe how to make a really big impact machine learning paper. And eventually here we have Sandy's machine versus man, and it turned out that machine won in that sense, at least. So I think that was maybe a good starting point. So please, the floor is open. So you can look at the picture if you need some reminding what was going on here. And Sandy, you get an overview, even if you were not here. Maybe I can start with one thing that maybe I've asked Robert. The cost of making these really huge new Google models when it comes to actually running them just in pure electricity, and the environmental footprint of that is kind of huge as well. Do you have anything to say about that?
Yeah, I think that's a big criticism. I think, I mean, it's not so much running them when they're trained, but you're right. During training, it's often a very big cost. And this has also been raised recently. I don't know if you've followed, so they have some trouble now, at least their image, Google, because of they fired the AI ethicist. And one of the things they raised in that paper that led to her firing was the sort of the ethical problem also of the energy use, right? So I mean, all of these is kind of opportunity costs that you take away when doing this. But I've also heard the counter argument that in some sense, knowledge and intelligence and better models all go together, and even a small advance in what we learn on a meta level about these type of building models could be in long term quite useful, right? So it is I think it's a quite difficult discussion to have. It's not easy to take one side of that argument, I think.
Well, when we're talking about arguments as well, High-Tech, you mentioned the war between statistics and computer science that's ongoing. So I mean, the Swedish solution here would be that that we would focus on the combination that we will, end up with after this day. We will, take each other's hand and say that the combination is always the best thing to do. So can you see that Swedish solution, Hitesh?
The nice compromise. I guess when I've seen this, so at the biostatistics meetings I've been to, I think one of the biggest problems has been stating what method you're using, because a lot of what people have called machine learning are essentially just statistical models anyway, and you could argue that yeah, so they're not necessarily the same, right? So a lot of these things that people are using are essentially statistical models in the first place. And I get the feeling that the problem here is how do you deal with uncertainty? And I think that's not really ever been handled, from my understanding, in machine learning community. They're not aware of uncertainty, right? So we talked about the uncertainty in the input and the uncertainty in the output. It's not clear to me. And so a good example is informative missingness, right? So in electronic health record data, sometimes when data is missing, that's actually telling you something. It's usually telling you, for example, in electronic health record data in the UK, the amount of data that's missing is actually related to your prognosis. The reason why you don't have any data is because actually you're in a bad state to begin with. You can't necessarily tease those things apart that easily with machine learning methods. It's not possible, right? You can't actually state that probability statement, I don't think. And I think that's kind of been the arguments a bit from what I've seen. It's about people rebranding statistical modeling and machine learning. That was one thing. And then this whole concept of uncertainty, which you can't really, from what I've seen, handle that well with machine learning methods.
Now, I think that this is also a big problem in the, let's say, in the differential equations-based models. And so these models will need input parameters as well, which are very rarely measured. And so the data is in general very poor. And what I think, and this is something that I'm interested in myself, but is that I'm interested in partial differential equations. And of course, these partial differential equations will be solved by, at least the solution will be approximated by numerical techniques like finite element methods. And the thing is that if you have a three-dimensional model, then of course these models will take a long time to calculate. So these simulations will be expensive. And what I want to do is I want to provide doctors with simulation tools. And in that sense, these finite element tools are not really effective for them. And so what I want to do is I want to train neural networks. I want to train artificial intelligence systems in a way that they can model these finite element models, and in a way that the doctors will have access to simulations. But of course, there is always a lack of data, and so I'm fully aware of that. So it's a challenging thing, I think. But I think indeed I'm pretty much in favor of the Swedish solution in a certain sense, that that you take the best of these two approaches, combine them.
Yeah, so
you can raise your hand if you want, or you can raise your artificial hand as well. Philip, please.
Yeah. No, I was just going to say, continue what Hitesh said about uncertainty. But I think that sort of mathematical biology or mathematical modeling, maybe in general, in terms of ODEs and individual-based models as well, that we've learned or we're learning to deal with uncertainty. And I think that Sebastian has made an important contribution to this, where he, in one of the models where they look at a mouse model, modeling growth dynamics of primary tumors, they actually have an error model for the data, which is very unusual when it comes to mathematical oncology. And I think this is where we need to go to think about about uncertainty in terms of what we measure, but also in terms of our predictions and standard error and confidence intervals and so on predictions. So yeah, I think we need more of that.
Well, thank you. Robert.
Yeah, so I have a comment on Hitesh comments there. So I'm not so sure. I agree with you that uncertainty hasn't been in focus, but I don't see anything fundamentally, over time. I think that will improve. And I don't see that when it improves, then these models automatically become statistical in that. So I agree with you that it's very important to be clear on what we mean and describe the technical details, of course. But I do think that you can do different things, which is not already clearly done in statistical modeling. And I think that that's the long-term thing we should focus on. So, I mean, you can slap sort of a Bayesian analysis on top of most machine learning models in some sense, and then you can handle some level of uncertainty. But so I agree with you, it's a current problem, but I'm not sure it's a long-term problem in that sense.
So, I mean, are they dealing much with missingness? So, I mean, I think you probably know the statistical sort of literature quite well. So when we think back to sort of Donald Rubin's initial papers in the 70s around about the different types of missingness, right? So you have missing completely at random, missing not at random, missing at random. I mean, how much of that thought is, I mean, I don't know the machine learning community that well. How much are they thinking about those concepts?
Well, I mean, increasingly, the point is that there's so much work, right? So we, I mean, I've never published in any sub area where, you know, you get 100 citations in a half a year, right? I mean, there's so much going on, and a lot of it is aware of lots of the statistical history and literature. So I do think that, yes, there are many sort of problems like any growing and young field, but I'm not sure it's kind of fundamental long-term problem. Yeah. Anyway.
Thank you. So I would like to let Mats Gisstrand say something here about the different kinds of errors that we were addressed. You know, the modeling error and the data error, if you want.
Yeah.
You didn't ask for the word, I just gave it to you.
Oh, yeah. I'm not sure if it's that what you're after, but I mean, we are quite specific when it comes to also modeling and trying to incorporate structure into the models that we are building in different fields in terms of error models of various kinds, ranging from observational errors to process errors to inter-individual variation and so on. And we also heard earlier today this overview or slide on pharmacometrics, which is an area where we're quite active, where it becomes more and more acknowledged that okay, maybe we have to at least turn into some other kind of equations than ordinary differential equations to describe some of our systems, because as more and more components and mechanisms enters that we cannot really capture mechanistically, maybe there's some other fudge factors or things that could be added to the differential equations. And one typical thing there is to do that is to turn into stochastic differential equations. Sometimes people think, okay, is that because there's a lot of unknown external or exogenous signals? That could be one way of, so to say, smearing out the process noise and say that it comes from there. But another pragmatic way of looking at process noise or the noise terms in the stochastic differential equations is also to, so to say, smear out all unknown mechanistic parts that somehow is captured in those kind of terms as well. And that is something that is slowly getting into the field of pharmacometrics, at least, and also in some other fields. I mean, in automatic control, technical applications where you're not talking about differential equations or continuous time in all kinds of processes, but also use quite a lot of difference equations. These things with process noise has been around for a very, very long time. And also people know the Kalman filter and these kind of things knows that this is quite old theory, but it enters more and more fields, both in a discrete and in a continuous fashion, I would say.
Oh,
So
thank you.
More about error models and uncertainty and also structure on those kind of things. I think that's very good.
Thank you, Mats. Sandy, did you?
Yeah, it was just to say that I think one of the issues that we deal with, at least in cancer biology, is that everyone's focused on reproducing and minimizing error in their experimental systems. So they're forcing the kind of results in some sense to be reproducible. And when you then take those experimental systems and then you say, let's try and translate that into patients, and now there's this diversity that's out there because not all patients are the same and they all behave differently. So you've trained your system on, say, data and mechanism that's in some sense artificial, right? And then, constrained, because that's the way reproducibility and statistical significance works. And you've kind of removed some of that noise or variance. And so I think one of the issues is teasing apart what's noise and teasing apart what's actually real process and mechanism that's driving a difference in the behavior, right? And how can we do that? And that's where potentially I think the synergy between mechanistic modeling and machine learning could really be beneficial.
Great. Thank you. I think it's time to leave the word to the man who started the day here, Bent Wennberg, see if he is happy about the answers he got to his 3 questions.
Yeah, I'm very happy and certainly happy about all the good talks I've been listening to today. Perhaps the first question I posed there of mathematical modeling being obsolete was supposed to be sort of not a serious question, because I hope not. I think it's not, but I think that new techniques are always welcome into the mathematical modeling community. And so I'd like to say thank you then to everybody who has participated in the meeting as speakers or asking questions or just listening to us. And I hope that we will be in contact in the future as well. Thanks a lot.
Thank you, everyone.
Thank you.
Thanks a lot. Take care. Be safe.
Take care.